Hello everyone, I'm Shamir Nasir. I'm a PhD student in Sam Martin's lab in Aberdeen. Today I'm going to present our work on some bits of our work package 3 work that we want to look at the multiomic data that we had from ATAC and CHIP to look at the regulatory regions and align the antiviral response in Atlantic salmon. So I'm going to repeat the experimental design again for work package 3, which I think it was already done by 3 people today already. We had both in vivo and in vitro approaches where we injected 6 fish with poly-IC and 6 PVS control and kept them for 24 hours to get the innate immune responses and then harvested head kidney. For the in vitro, we first extracted primary leukocytes and then stimulated those primary leukocytes in 6 well plates and then made these assays, extracted RNA for RNA-seq and then made ATAC and CHIP libraries. We only made 2 CHIP libraries. We used H3K27AC as an active mark and H3K27Me3 as a repressor mark. So after sequencing, we identified around 53,000 unique ATAC reads and 72,000 the active mark, histone active mark, and then around 68,000 for repressive histone mark. And then if we did differential analysis, we had 600 ATAC up and 48 down. And around 1,500 the active histone mark up and then 1,200 down. We also had 795 down H3K27 and 1,200 up and down. So the RNA-seq we had around 1,400 genes upregulated and 1,000 downregulated. This is already published, and Thomas talked about it yesterday, where we compared Atlantic salmon and rainbow trout in vivo and in vitro antiviral responses. So we wanted to see if our data from different assays overlap. So we did see some overlap between H3K27AC and H3K27Me3. There were 21 regions that were overlapping between them, which is a small number. We expected that. We also had 10 between RNA and H3K27Me3. But we had majority of our peaks, we had around 200, were only shared between ATAC, H3K27AC, and had RNA-seq. We also had 171 regions that were in ATAC-seq and also showed H3K27AC, but were not present at gene level, so they were not transcribed. So I think it would be interesting to see our microRNA data to see if we find any answer for that. So I was interested in these 197 genes, which had open chromatin using ATAC, and then H3K27AC, and then RNA-seq. So we did some gene set ontology enrichment analysis. So these were the GO terms that were enriched. They were all to do with antiviral responses. We had innate immune response, we had response to viruses, and we had some ubiquitination. And in general, it was indicating that we had those genes are involved in antiviral response and the activation of innate immune system. So next, I want to see the pathways that are involved in the antiviral responses. So induction of interferon-stimulated gene, which ISGs, which are the key antiviral genes. So I picked two pathways, RIG1 pathway, which is this pathway detects viral RNA, and then initiate this pathway where transcription factor IRF7 and IRF3 binds to the promoter regions of interferon type 1, and then interferon type 1 are induced, and then these interferon type 1 binds to the interferon receptors and initiate the JAK-STAT pathway, which in this pathway is initiated, and then this complex, STAT1, STAT2, and IRF9 is formed, and this complex binds to the binding site of ISRE, interferon response elements, and then ISGs is induced. This is a conserved region, and it's involved in the induction of ISGs. So we wanted to see if we can find the ISRE regions within the ISGs. So I picked here, just showing the STAT FAT3 genes that are involved in this complex, in the JAK-STAT, STAT1, STAT2, and IRF9. So we can see that I've only used one paralog for each. You can see that, so here, this is I picked the longest transcript, and the arrow is showing the direction of the sequence. And then this is the RNA-seq data, where we have the PBS and poly-IC stimulated. And on the top, we have the ATAC data, PBS, which is control, and poly-IC. We do see a clear differentially increased peak in poly-IC compared to the PBS. Then we have the H3K27IC. Again, we see differentially increased peaks in poly-IC stimulated fish. And while for the repressor mark, we don't see any peak at all for the poly-IC, but we do see a base level peak for the PBS. And then we found these ISRE sequences exactly lining on those peaks. For some of them, we had more than one regions of these motifs. So I call these functional motifs because they were on the open chromatin regions. But just to note that these sequences are everywhere in the genome. So I'm not sure the other ones that are at the promoter region, but they don't have a peak for ATAC or for the chip. I'm not sure if they could be called functional. So I've also done this for the RIG1 pathway, but I'm not going to show it here. So next, I wanted to look at these motifs, see if we can see them at see difference in paralog level. So this is MX, which is a key antiviral gene. It has 7 paralogs in salmon. And only one of them had ATAC and CHIP differential peaks. And while we have 2, which were differentially expressed at RNA level, but this one didn't have any ATAC or CHIP peak, and also, of course, didn't have any ISRE region enriched as well. But only this one, which had ATAC and CHIP peak for the CHIP active mark and ATAC, while at 2 ISRE regions. So I've done this again for lots of antiviral genes. I'm just showing one here. Next, we also did some transcription factor binding site analysis using ATAC and CHIP data. And again, we have a ISRE motif enriched binding site for the IRF9, which is an ISRE sequence, was highly enriched in poly-IC stimulated fish. We also had BCL6, which is a B-cell differentiator. The AP2 is an AP1 binding site family. and IRF6 as well. Yeah, we have lots of epigenetic data from ATAC and CHIP, but obviously we don't have time to present all of them, so I picked a selective one. To conclude, we were able to investigate antiviral response gene using multiomic approach. We were able to identify regulatory motifs involved in antiviral response, and we also found parallel-specific functional ISRE regions in key ISGs. I want to thank everyone involved in this project, especially AQUA-FAANG, for giving me the opportunity to do my PhD, and also my supervisor, Pierre's lab, and also Dan's lab for the help.